So thank you everyone for joining us today. So this is the Master the Art of Science Writing webinar, and this is brought to you by the British Ecological Society in partnership with Wiley. So these are our British Ecological Society journals, if you haven't seen them before, and I'm going to be talking a little bit more about us. So who are we? I'm Rowena Gordon. I'm the Senior Managing Editor at the British Ecological Society, and I manage two of the journals, so I'm in the publishing team, and I have a background in ecology as well from Queen Mary University of London. So I'm just going to be doing the introductions today, and then I'm going to be handing over to Professor Emma Sayer. So Emma Sayer is one of our senior editors at the British Ecological Society on the Functional Ecology Journal, but that's just what she does in her spare time. In her main role, she is an ecosystem ecologist at Ulm University, Germany, and also Lancaster University in the UK. So she is a senior editor for us, and she is really, really fantastic science communicator and educator. So I'm going to be handing over to her shortly, and she will be doing the main parts of the session today. I'm just going to give you a quick intro to the British Ecological Society, and then I'll hand over to her. So who are we? We are the British Ecological Society. So we are the oldest ecological society in the world, founded in 1913, and we are a global community of ecologists, and that's ecological practitioners and ecological researchers. We have 7 international journals, and obviously I'm from the publishing team, so I think they're the best thing about the society. But one of the nice things is everything that we earn through our journals is invested back into the ecological community. So some of the things that we do as a society is we have an education and careers program. So we provide free resources for teachers, and we run summer schools that it's free to attend for children and young people. And we're just trying to engage the next generation in ecology with this program. We also have grant funding. So we give out about £500,000 British pounds in grant funding every year. And 51% of that grant funding is ring-fenced for the Global South. So even though we're British in our name, we are global in the way that we work. We do actually also have a round of grant funding that is open for application at the moment. So there's a little bit of info about that in the break, but I do encourage you, if you're after some grant funding, to have a look at that on our website. We also run events and communities. So we run Europe's largest ecology conference in December every year. And this is a really fantastic event. That's Yadvinder Mali, who was our past president, speaking at a recent annual meeting. And I do encourage you that we have online versions to join that as well as in person. So I do encourage you to engage with that, because we have a really large community of ecologists. It's a great way to network. And we also have special interest groups, which is little groups that you can join to find other people with the same interests as you, and those are global groups as well. And finally, we do policy work. So we have a small policy team based in-house, and they're supporting the government to make recommendations backed by the best ecological science. So as a publications team, we work with authors to ensure that the best possible ecological science is published, shared and promoted. So again, we're very global. Our editorial boards have editors from 48 countries, and our authors come from all over the world. We have a friendly and rigorous peer review process, which authors have told us helps make their paper better. And post-acceptance, we work with authors to share their work with as big an audience as possible. So I've included there just an example of one of the blogs that we had recently. about fire ant invasion in plantation forest. But we also do promotion on social media platforms, podcasts, videos, and events. So that's what brings us here today. So one of the other things we do as a publications team is we run free capacity building workshops like these, which is just aimed to help the ecological community to ensure that we're publishing the best science and the best science is out there. So some of the other workshops we've done in the past are how to be a good reviewer, how to become an associate editor, and how to promote your research. But today, it's all about mastering the art of science writing. Okay, so here's the schedule for today, and this is in Beijing time. So I'm going to be handing over to Emma Sayer very shortly, and then you'll see there's a break at 4:30 p.m. Beijing time. And that's a 15-minute break, and then there'll be another break. There'll be at the end of the session, I'll be coming back just to close off, and we'll be finishing at 6 p.m.
So yeah, good morning, everyone. My name's Emma Sayer. As Rowena's already mentioned, I'm a senior editor at Functional Ecology, and I'm really, really interested in how to improve the way we communicate science and to make scientists better communicators. Today, the webinar is split into 2 sessions. And in this first part, I'm going to be talking mostly about how we look at and how we think about the different sections of papers. So how do we go about ensuring that papers fulfill their intended purpose? And this might sound like an odd place to start when we're talking about science writing, but one of the really important things to remember is who we are writing for, why we are writing scientific papers. And many of us forget this because we're very focused on, I need the paper for my CV, I need the paper for my career, I need the paper because my supervisor has told me so. And we forget that actually we are writing to communicate our research with other researchers, and that we need to think about how we give the audience, give the readership what they want. I'm going to start talking a little bit about how we, about the purpose of scientific papers, research papers, and then in the second session, I'll mostly focus on some tips and tricks that you can use to structure the text, create a strong narrative, and make sure that your paper is easily accessible, easy to read, and easy to understand. And the tips and tricks, the techniques I'm going to show you in the second session are really important when you're writing in English as a foreign language. It's a big hurdle. I have huge respect for people writing science in a foreign language, especially when you're even using a completely different alphabet. And I know that this is often a big worry to people that they get their English right. And actually, the really important thing is if you can structure the text well and give the reader the right information in the right place, then the smaller grammatical errors and language errors don't matter. They're not important anymore. So that's what I'll focus on in the second session. I also wanted to flag up some the sources of a lot of the information I'm going to show you today that are also useful resources. So I've already written several editorials for Functional Ecology on different aspects of science writing, and this webinar is mostly based on those 3 editorials. If you're really interested in learning more about science writing, I think this writing science by Joshua Schimmel. This book is probably the best resource with lots and lots of exercises and more information. So I wanted to start by talking about how or what types of papers the British Ecological Society publish, and what types of papers other top journals in the field want to publish. And that is quite simply papers that advance the research field. And this is a really important thing to remember right at the start of the writing process. So an advance is not necessarily just that something is novel or new. An advance actually demonstrates, for example, important areas of research for future studies. It provides completely new perspectives on a phenomenon, or might even reveal a new phenomenon. And of course, that motivates other researchers to fill knowledge gaps with new studies. It might also be that a paper facilitates or promotes future work by exposing issues or presenting improved methodology. So, for example, if your paper shows a problem with existing methodology, or presents a new method that people can use. So basically, a paper that advances the field leads to research, to new research, or to changes in the way we do things, changes in policy, or changes in practice. So how do you demonstrate the advance to the field in your paper? Well, the first thing is to ask yourself why a researcher from another country or another research field should read your paper. What might be interesting to them? Then, what does your paper demonstrate that others haven't shown yet? What is new and interesting? And then one of the most important things is to think about how the research community will benefit from your paper. Why should somebody read your paper? Why might someone cite your paper? And of course, when we think about why we cite papers, it's usually because somebody has given us something that is really useful for our own research. So thinking about what aspects of your study might be cited by others is a really good way to start. And then we want to demonstrate the scientific advance in several sections of the paper. So the abstract, it's important to demonstrate how the research advances the field, because this will motivate people to read the paper in the first place. In the introduction, the advance is often the main justification for the study, and I'll come to that later. Then in the discussions and conclusions, the advance is often the key take-home message of your paper. So if there's one thing that you want people to remember from your paper, what is it? It should be this scientific advance. So this is I've already started talking about individual sections of a paper and how to demonstrate the advance, how to build up a narrative motivating people to read your paper. And this brings me to the purposes of these different sections of the paper. So a classic research paper will obviously have an abstract, but the main sections are introduction, methods, results, and discussion. And what's really important here is that all of these sections, as shown on the diagram here, are informed by the research question, the aims of the study, and the hypotheses that are posed. And this is absolutely essential when you're writing, that all of these sections are linked by the aims and your questions or hypotheses. So the research aims really guide the entire writing process. The introduction, for example, provides the context and the background for the research question and all the information needed to justify the hypotheses. Then the methods demonstrate how the design of the study and the different measurements, the different variables are going to answer the question and test those hypotheses. The results provide empirical evidence that will help us answer the question and test the hypotheses. And then in the discussion, you are showing how the results answer that question, meet the study aims, and of course, you're placing your hypotheses and this evidence then in context. So your discussion then links back to the introduction, because you're placing the results in context and demonstrating how you have tested your hypotheses and answered your questions and met your study aims. So this is a really, really important point to remember right at the start of the writing process. The study aims and the hypotheses or the research questions must be absolutely clear. Otherwise, you can't create a good narrative for your paper. So obviously, I've said questions or hypotheses now several times. So when do we have questions? When do we have hypotheses? And in general, it's always better, almost always better, if you can formulate hypotheses, because that's how science works. We generate hypotheses and we test them, and we attempt to reject the hypotheses or reject the null hypothesis. So a good rule is that if you have very clear expectations of what your results will look like, the outcome of the study, then you have hypotheses, or you should formulate hypotheses. So there's enough existing information for you to say, okay, when I measure this, I think this is going to happen. You have an expectation, that means you have a hypothesis that you're going to test. On the other hand, in some cases, you might work on a very novel topic. There's very little information about this. It's really new to the field, so you don't know what to expect. And in this case, you're probably working with questions rather than hypotheses, simply because there isn't enough information to formulate those expectations. So it's better to formulate hypotheses if you can, but what is really important when you're formulating hypotheses is that they must be specific and they must be testable. So I already have a question for you. We're going to take votes on how to formulate a specific testable hypothesis. And all I want to know is which of the two hypotheses is specific and testable. So the research question is, how is beetle abundance influenced by edge effects in tropical forests? Hypothesis 1A is beetle abundance will change due to edge effects, and hypothesis 1B is that beetle abundance will increase with distance to the forest edge. So essentially, specific and testable hypotheses, you can recognize them because they usually include the magnitude and the direction of the expected effects. So in this case, yes, we might expect beetle abundance to change due to edge effects, but we can't test this hypothesis, not properly, not rigorously. So just saying something will change is not a strong hypothesis. Instead, we need to test a direction. So in this case, an increase, and we need to say how we're going to, or we need to indicate how we might test that. So we need a distance to the forest edge to test these edge effects. And this hypothesis now gives us a really good indication of what measurements we're going to do. We're going to measure dung beetle abundance. We're expecting dung beetle abundance to increase along, for example, transects away from the forest edge. So this is a specific testable hypothesis. So this is a great starting point. But of course, we have all of these different paper sections. to think about how we get our message across in all of the different paper sections. And I'm going to start with the abstract, because the abstract is often, although it's often the last step of the writing process, it can also help to write an abstract right at the beginning before you've actually written the paper. So when you write a draft abstract at the beginning of the writing process, it can actually help you structure the paper. Often because it's so short and so concise that you need to focus on key messages and the most important information. And that can really help get the structure across in your entire paper. Then, of course, once you've written your paper, you want to rewrite the abstract or revise the abstract to make sure that you really have covered those key points, because things can change during the writing process. Now, I personally find it very hard to be concise. I find it difficult to describe an entire paper in 250 to 300 words. So what I often do is I'll start with a much longer text, and then I will cut. I'll cut anything that is non-essential, anything that is tangential, until I'm really covering the most essential points and fitting to the word limit. So this is a good way of starting and making sure that you know what your key messages are before you write, and that those are very clear in the abstract. And this is important because the abstract is like the advertisement for your paper. So you're advertising your paper to people and hoping that they will read it. And the abstract is often the only thing that people will read. So if you're unlucky, it's the only thing that an editor or reviewers will read. So it really needs to contain the context, the background to the study, the relevance of the study, and the most important take-home messages, because that is what will motivate readers to keep reading the rest of the paper. It's also, when you submit your paper, the first thing the editor will read, and often the only thing some editors will read. They will make a decision, a first decision, about whether your paper is a good fit to the journal based on the abstract. So you want to make sure that the abstract contains all the key points in an easily digestible form, and that is usually a very brief background or justification for the study, so providing the context. Then a broad description of what you did, not too many methodological details, but an idea of what the study entails. Then the main findings, and a final statement or synthesis about the relevance of the study. And the BES journals have a very nice structure in which the abstract is divided into 4, maybe 5 paragraphs. Here's an example from Functional Ecology, where we have a brief background providing the context for the study, then the broad description of methods, the main findings, and the relevance. And I'm aware the type here is quite small, so you can look at that in your copy of the slides after the session. But essentially, the brief background shows what the problem is, what the context is, and where is the knowledge gap. So in this case, this is about bottom up and top down forces. So plant defenses and natural enemies, regulating herbivores, and the fact that it's unclear how much complementarity exists between these different controls of herbivores. Then the broad description of methods outlines laboratory and greenhouse bioassays, and presents the study species, or the study taxa, so herbivorous mites and predatory mites on mutant tomato plants without defensive hairs. So that already gives us a really nice broad description of what the study is without lots of detail. Then the main findings are presented, and the relevance, there's a very nice synthesis here. In just one sentence, the authors write, our results lead to the counterintuitive insight that removing instead of introducing plant defense traits can result in superior protection against important pests through biological control. So this is a very elegant way of summing up the key points of the study and demonstrating why it is novel and why it is relevant and important for future research. So this is a very nice example of a well-written abstract. Then, of course, the title is also advertisement for the paper. And the title should hook the reader and want them to read, or make them want to read the abstract. And I actually do, or I often, with my students, I'll do a whole session just on titles, abstracts, and keywords and making sure they match. But the essence of the title is that it should be brief and clear. It must say what your paper is about, and it should ideally summarize the main finding of the paper. Very often, as an editor, I see very long descriptive titles that tell me what the paper is about, but don't tell me why the paper is interesting, why it represents an advance, or what the main finding was. And those papers, often I will actually ask the authors to, if the paper is of interest, I'll ask the authors to rewrite the title to include the main finding. So we're going to have a second poll about titles. Here you have two options. Title A is that litter addition enhances early stage decomposition in a lowland tropical forest, and title B is effects of litter addition on early stage decomposition in a tropical moist forest. So which one do you think is the better title? Which one contains the essence of the study and the main finding? So title A or title B? So most people think title A is better. I would agree with that. So title A is just that little bit more specific. So title B tells us that there are effects of litter addition, but it doesn't tell us what those effects are. So that small change, litter addition enhances early stage decomposition, is quite important because it tells us the main result. And in a way, this is very similar to what I was saying about hypotheses being specific and testable. Magnitude and direction of the effects are also important in the title. So that small, subtle difference between these two titles actually makes a big difference to whether it really attracts readers and represents the paper in a good way. So a good title tells you the key findings, and it has a strong take-home message. So it should be when people remember the paper, they already know what the main finding was. And this second title, like I said, has a weak take-home message because it only suggests that there's an effect, but it doesn't tell us that key finding. So moving on to the introduction, you know, we've already got a working title. We have a draft abstract of our paper, and although we might not write the introduction first, the introduction is the first thing that people will read, the first part of the paper that people will read. And the introduction is there to provide a background and a rationale for the study. So why are we doing the study? Why is it interesting? And a lot of people, myself included, struggle with, or have struggled with, writing the introduction, because it seems there is so much context, there's so much background, there's so much literature on the topic. How do I narrow it down to create a nice introduction to my paper, to my study? And essentially, we need to think about the introduction as setting the scene for our study. So we're giving the background for the study, we're telling the reader why they should be interested, and we need to do this in a logical train of thought that leads the reader to the conclusion that the study is novel, exciting, and worth doing. So by the end of the introduction, we really want the reader to go, oh, I need to read the rest of the paper to find out what the results are. And that is often, or might seem, often seem quite difficult, like I said, when there's so much information out there. But actually, for the introduction, you don't want to include everything. You don't want to include everything you know or want to say. It's very tempting to review the literature, read the literature and go, oh, this is really interesting. I need to put this in my paper. But the introduction is not a review of the literature. And it's also not a place for methodological detail. So if you're writing a methods paper, then your introduction will be largely about methods. But for most research papers, we might only have a sentence saying that we did a field study and maybe the location, but we don't really want more than that in the introduction. So the introduction is not a place for details on methods. Instead, you only really need 4 paragraphs. The first paragraph should outline why the topic is important in the broadest sense. So this might be about the importance of the ecosystem, the importance of a process in understanding the impacts of global change. It really is broad. Then in the second paragraph, we want to narrow the subject down to give the scientific context. In the third paragraph, we're showing the issue, the problem, maybe the key knowledge gap that we want to address with our study. And then in the fourth paragraph, we have why, the study's novelty, so why we're doing this, how we're going to address the knowledge gaps. And then we end the fourth paragraph with aims and hypotheses. And I should say at this part that, of course, it's possible that all 3 of the first paragraphs contain important knowledge gaps. But in the third paragraph, we really want to focus on the issue we are going to solve with our research. And then the fourth paragraph focuses on the study itself. So what we're doing here is narrowing down. We're starting at broad and we're successively narrowing down to our specific study. And this is something called funneling. It's a technique called funneling. And I always say, start big, finish small. So start broad, general, wide context, and finish narrow with specifics, details, or complexity. And this is an absolutely essential technique, and you're going to hear start big, finish small quite a few times during these 2 sessions. So because this technique is unbelievably powerful. You can use it in science writing. You can also use it in reports. You can use it in application letters for jobs, all kinds of settings. It's actually a journalistic technique in which you are giving the reader the information they need to understand the next level of complexity or the next level of detail. So this is a really important technique to remember. And as I said, I'll keep coming back to it. So always starting broad and narrowing down to specifics. And I'd like to show you a brief example. Don't worry about reading this. This is also from one of our journals. This is from Journal of Applied Ecology. And you can see here that the introduction really is just 4 paragraphs. A couple of the paragraphs are quite long, but it is just 4 paragraphs. And these 4 paragraphs, I've just highlighted the sort of the first few sentences of each paragraph here and colored them by topic. So the study starts out with the global context, so talking about eutrophication in densely populated areas. So it doesn't even say which area of the world. This is global. This is a major problem, eutrophication in, for example, urban areas. Then the second paragraph talks about high internal loading of phosphorus from lake sediments. So this is one aspect of eutrophication. So now we are narrowing down to the scientific context. So we've gone from eutrophication everywhere to phosphorus in lake sediments. In the third paragraph, the authors talk about lake restoration techniques to combat eutrophication. And then they refer specifically to phosphorus loading. Then in the last paragraph, so we now have, this is the issue. There are lots of different restoration techniques. We don't know which ones work best. So the study aim in this case is to give an overview of these different types of restoration projects and to evaluate their results. So what we have here is a nice example of funneling. And even within these paragraphs, you can see how we've gone from here in this first one, we have gone from broad global to European in just a few sentences. So even within the paragraphs, you can use this technique to create a nice narrative. So we go from global eutrophication to European eutrophication to phosphorus loading. And then finally, in the final paragraph, we see that we're talking about Denmark and the Netherlands. So actually just 2 countries. But the results will link back to, as it says here, the context of international restoration projects. So again, this is a really nice example of how you can use this funneling technique to demonstrate the relevance of a study to the reader. And in this case, the authors have shown us why a study in just Denmark and the Netherlands actually might have global relevance. It might be much more widely applicable. So what you can do when the next time you want to write an introduction, or you're thinking about planning your introduction, is quite simply to start to create an outline for your introduction in 4, maybe 5 bullet points. So just single points that give the global context, the scientific context, the issue you're going to address, and then the aims of the study. And then you can use those points as a guide to write the rest of the text. Now, of course, this is all sort of rule of thumb, and it's possible that you might need to be flexible. So you might need to combine paragraph 1 and 2 into a single paragraph, or paragraph 2 and 3. It might make more sense. And if you have a complex study in which you're looking at many different aspects, you're looking at maybe interactions between different taxa or different ecosystem components, then you probably need a 5th paragraph to introduce a new topic. But overall, the structure remains the same. So global context, narrower scientific context, the issue, and then how the study is going to contribute to knowledge on that issue. So that's all I need to say to introductions. It is easier than this finding out about this 4-paragraph introduction structure really helped me focus my introduction and my writing in the introduction. And of course, towards the end of the introduction, we're giving the study aims, and we might give a very brief overview of the study site and the method that we're using. So the design of the study, field study, lab study. And that then immediately leads us to the methods section. And I always like to think of the methods section as kind of a cookbook. It's the recipe for my study. And this is a good way of thinking about it, because if you think about a recipe for a nice dish that you're preparing, you want to be able to follow that recipe step by step and get the same result each time. And so this is really an important aspect of the methods. And the difficult bit about the methods is getting that level of detail right, making sure that you have this recipe for your study. So the methods must provide enough information for the reader to understand how the study design addresses the research aims, tests the hypotheses, answers the questions that you've given at the end of the introduction, and of course, whether the methodology and data analyses are appropriate to do that. So is the methodology, are the variables, and are your analyses appropriate for testing your hypotheses? And the recipe aspect of this is reproducibility. So the details that you provide in the methods, the description of your study, should allow the reader to recreate each step. So if someone else were to go out and do the same study at the same location, they should get very, very similar results to you. They should be able to recreate the study the same way that we follow a recipe to recreate a dish. And this is very, very important. So we need to include any details that might influence the way the data are collected or the amount of data collected, the approach we use to analyze the data, so the statistical tests or models, and of course, anything that could influence the results and their interpretation. This is very, very important. So some example of really crucial details are the sampling, a description of the sampling sites and the experimental treatments. And that might include the type, number, and size of the sampling units, obviously the level of replication, and then of course the number of samples, the frequency of sample collection, and then other details such as the instrument that you used, the amount of sample that was then used for further analyses, and so on and so forth. And of course, how the data were handled in the data analyses. So these are absolutely crucial details that you really must give in the methods. And you can ask yourself, does the method section really give the reader a mental picture of your study? And could you draw a figure or diagram of the study design or the procedure based on the description in the text? And this idea of creating a picture is quite important, because we must remember we've probably worked on this study for a long time. We're very, very familiar with everything we've done, but we need to describe it for somebody who is seeing this for the first time. So as a reviewer, and sometimes even as an editor, I might read the method section and I cannot understand what the authors have done, and I will try and draw a diagram to see if I can work it out based on the text. If I can't do that, then I immediately say that I can't evaluate the rigor of the science, because there isn't enough detail in the methods, or the methods description isn't clear. So this is very, very important, because it is one of the main reasons for rejection post-review, is if the methods are not suited or suitable to test the hypotheses, or if the method's description is so insufficient that the reviewers cannot judge the rigor of the study. So at the very least, you would be asked for major revisions, maybe even reject and resubmit if it's not possible to really understand how the study design answers the research questions or tests the hypotheses. This is very important. So another thing you can also do if you have a complex study is at the end of the method section, or at the end of the relevant sections, you can summarize sites, sampling units, treatments, and replicates. You'll often see this as a statement saying, in summary, there were, I don't know, 20 plots with 5 replicants and 4 treatments. Each plot was split into 2 subplots with these subtreatments. And then you have a nice sort of overview of the study design if you have something more complicated. And again, this concept of starting big and finishing small is important in the method section. So if you start, and you'll see this in so many papers, we usually start with a description of the study site, the climate, maybe the soil type or the vegetation type, whatever is obviously relevant to the study, but we start broad. And then we'll build in, for example, transect, plot, sampling units, and treatments. Then we'll start talking about sampling for individual variables. So we tend to do this, start big and build in details or complexity in the methods. And you can really use this to structure the methods and make sure that it's easy to follow what you did and why. It's also very useful if in the methods you refer directly back to your hypotheses. So that you say, to test this, we measured that, or to determine this, we measured that. To actually give the purpose of the methods in each case. That can be very useful for the reader to understand how your study design tests your hypotheses and answers your questions. So the next section in the paper is the results. And what's important to remember with the results is that you're providing a biologically meaningful factual report without interpretation. So you're reporting what you found from your study without interpretation, but you need to do so in a way that relates back to the context of the study and its biological or ecological relevance. And again, it's really good to remember how your results link directly to your questions and hypotheses. And I find it very useful here to differentiate between the key results. So these are the most novel findings that address your study aims. So the most interesting results, if you like, and they will also be the focus of the discussion. But then very often we will have many supporting results. These support the key results and help with the interpretation of findings, but they're maybe not very novel and they won't be the focus of the discussion. One example I like to give here, I do a lot of work on soil carbon dynamics. I measure soil respiration in response to different treatments, for example, and I really need to include data on soil temperature and soil water content, because we've known for over 50 years that soil moisture and soil water content and soil temperature influence CO2 coming from the soil, so soil respiration. And we've known that for a very long time. These results are probably not particularly interesting. What is really interesting are the CO2 results, but I have to be able to show, for example, that temperature and soil water content differ or don't differ among my treatments to be able to interpret the differences in CO2. So in this case, my key results will be the big changes in soil CO2, and soil temperature and soil water content will be supporting results that I use to interpret my data on CO2. Now, one of the big issues with the results section, especially among ecologists, is that we tend to get lost in statistics. So there is a very big focus on statistical testing and modeling and providing significant p-values. And it's really important, if we want to provide a biologically meaningful report, and we want the readers to be able to understand what our results mean, we need to remember that we're using statistics to support the ecology. So we use statistics to support statements in the results. We're not using statements to support statistics. So again, the ecology needs to be at the center of the results. And another thing to remember is again, you know, the hypotheses should give the magnitude and direction of effects we're expecting. So our results also need to give the magnitude and direction of effects. So instead of saying X and Y were significantly different, we might want to say X was 15% higher than Y. And then we give our statistics in parentheses after the results. So if we make very clear statements about the ecological meaning of treatments, patterns, and effects, we can help the reader understand our results, and also the reader will start thinking about the results and interpreting the results as they read. And obviously you would like them to come to the same conclusion as you do in the discussion. So using these clear statements, biological statements, about your findings, your results, is quite important. So I want to show you here two completely different, or they're actually the same results. Here I've written them without any statistics to make them clearer, and you can look at these on your own time at home. But essentially, the first text uses statistical terms, the second text uses real world terms, and I wanted to show you this as an example. Here we see, for example, the statistical terms. We have things like significantly positively correlated with, significantly negatively correlated with, marginally significantly correlated with. Now, this is not only hard to read, but it also means that the reader has to think for a moment, even if it's just a second. Significantly positively correlated, that means increases with. Okay, and it's that second, when you have to do that with each sentence, you have to take a moment and go, oh yeah, that means an increase, that means a decrease, that means this increases and that declines. That is actually already a barrier to understanding. So instead, we use words like declined with, increased with, and here you can also see I've given an example, a non-significant trend towards lower foliar NP ratios, and that can help the reader create a picture of the results in their mind. What I've also done here is these are quite complex results in this case. I've actually started this section of the results with a summary statement that provides context. So here I've written, interestingly, foliar NP ratios were influenced by shifts in the concentrations of both N and P, depending on the global change driver. Now, what I've done there is I've summarized this section already, and I've signaled to the reader that actually this is going to get complicated. So I have changes in nitrogen, I have changes in phosphorus, and it depends on which global change driver is having an effect on foliar NP ratios. And that means that the reader is now prepared for me talking about changes in nitrogen, changes in phosphorus, and different global change drivers. So I've provided the reader with the context they need to understand that complexity. So again, I started big and I'm building in the detail and complexity. So you can try this at home to improve the readability of your results. So start each paragraph, for example, of the results using what we call a topical sentence that provides context. So this topical sentence basically tells the reader, this is what's coming. This is what I'm about to talk about. This is sort of like, this is the crucial information you need to know. Then, as you're writing your section, always remember to give the magnitude and direction of effects, use biological terms, and then build in the statistics and numbers. And what I like to do, because I'm just as guilty as everyone else of focusing too much on the statistics, what I like to do and what I always tell my students to do is to make figures. So always look at your data, always look at the figures first, even before you start analyzing, and then also write the first draft of your results by referring to those figures. So without the output of statistical texts, can you see your results in the figures? What do you see? And then write your results based on that, adding in the statistics and numbers later. And what's important here, and what I always tell my students, is if you cannot, if you have a statistically significant result that you can't see in your figures, you have a problem. So either your statistics are incorrect or your figure is not effective. It's usually the former. It's usually that your statistics might need revising, but it could also be that your figure does not show the result to the best effect. So this is one of the most important aspects of the results. Your figures should show each result at a glance. So the reader should be able to look at a graph like this one and see, oh, the number of publications declines with the amount of beer that is consumed. So in this case, we don't know that whether researchers drinking more beer produce fewer publications, or whether they drink more beer because they don't have many publications, but we do see in this case that there is a very clear correlation between the two. So figures should show your most important results at a glance, and tables are best used for things like summary data or site characteristics. Most people will find it much easier to interpret a figure than a table. And one tip here is that if you have a section that reads like a very long list of numbers, then you probably need a table instead. So tables are great for sort of overview of characteristics and summary data, figures for the key results. And there's a nice paper in PLOS Computational Biology that outlines the 10 rules for effective figures. I've picked out the 7 most relevant ones to ecology here. The first is putting yourself in the reader's shoes. How can I show the reader what I need them to see? The second is then, obviously, you need to identify your message. What is important? What do I need to show? You need to provide very clear self-explanatory legends, and I'll come to that in a moment. Use colour and symbols effectively, for example, to highlight differences between treatments or sites. It's obviously very important not to mislead the reader. So you want the reader to see your results at a glance, but not manipulate the reader into seeing something that isn't there. Avoid clutter. So I've seen some really, really beautiful figures with lovely colours and lots and lots of detail, but I cannot see the result. And that means the figure is too cluttered. I need to be able to see that result, that difference between treatments or sites, or that relationship between variables. And that means that basically the message to the reader is more important than the beauty of the figure. So you can have the most beautiful figure, if the reader can't see that key result, can't get the message, then you've created an artwork and not an effective scientific graph. So one important aspect to remember when you're creating your figures and writing figure legends is that a lot of people will not read the results text. They will look at the figures and work out the results from the figures. So your figure legends must be understandable to somebody who hasn't read the paper, who hasn't read the text. They're just looking at the figure and the legend. And what I'm showing you here is quite a funny one. This is actually a real figure out of the European Urology Journal from 1993. We have a picture of a rat with a rather strange underpant, and all the legend says is the underpant worn by the rat. Now, at this point, everybody is thinking, why is the rat wearing an underpant? And is it important that it is a specific underpant? And all of these kind of things. So what on earth is this rat doing? What are the researchers doing to this rat? Loads and loads of questions. We have no idea. We cannot interpret this figure based on the information provided. So here's another example. This is a very simple box plot, where we can just take a moment to think, what do we need to know to understand this figure? So if I show you this figure, what information do I need to supply so that you can understand and interpret this figure without referring to the text? Now we can see across the top, I have abbreviations, L+, CT, and L-. I have methane flux on the side. I have two different colors. There are obviously the box plots. There are also some circles and the x-axis week of the year. And I've got these different top and bottom panels. So one of the important things is, where is this study? Where is the methane flux from? Where is this located? When was it carried out? So I've given week of the year, but actually, you know, unless I really, really think, I don't know what season this is, I don't know what month this is, all of this is quite important. Then I need to explain all abbreviations. I need to explain all of my symbols, and that includes what the box plots delineate. And then I need to give things like means, errors, and sample sizes. And then any other key information that the reader might need to be able to interpret this figure. So here is actually the legend to this figure. So we have soil methane fluxes. That's the first sentence. So now we know methane fluxes are coming from the soil. Under individuals of two common tree species, we have Hysteria cancina, red top panels, Simera aruba amara, blue bottom panels. So there we have our colours. In experimental litter removal, control and litter addition treatments, and there I give the abbreviations, in a tropical forest in Panama, Central America. So location of the experiment, measured weekly from March to July 2014. And then I've also put during the transition from the dry season to the wet season. Then right at the end, ranges, so boxes and whiskers, and median lines are shown for n equals 4 individuals per species in treatment. What is missing here is that circles denote outliers. So if we go back to the graph, the only thing missing are these little circles, that they're outliers. But what I wanted to show you in this figure is that I mentioned the transition from the dry to the rainy season. And if you look at the graph, you can see that there's a very clear pattern in these methane fluxes. Everything is below the 0 line up to about week 20, and then above the 0 line from week 22 onwards. This is my transition from the dry to the rainy season that I describe here in the legend. So I'm using the legend to direct the reader's focus to this transition. And of course, in this case, we could have done it even better by shading, for example. So having 2 different shades, wet and dry season, that would have made it even more obvious. But the important thing here is that the legend explains everything I can see in the figure. I don't need to read the results section. And if you see what I've done here, I've started with a sentence that summarizes exactly what the figure is about. So soil methane fluxes under individuals of 2 common tree species. And then I've successively built in detail, ending with the things like sample sizes and what all the other symbols are. So in this case, what the boxes and whiskers are. So essentially, I've done the same funneling technique here in my legend as I've done in the rest of the text. I've started big, I provided a topical sentence, and then I've built in complexity and detail. So I've started big, finished small, even in the legend. So that's all I wanted to say about the results section and presenting the results. Obviously, once we've presented the results, then we need to discuss them. And the discussion is often the hardest part of the paper to write, because it's in a way, it's the least technical. So the methods and results I often find very easy to write, very straightforward, because I know what I've done, I know what I found, I know which results I need to focus on, but then interpreting them is another matter entirely. But interpreting the results and putting them in context in the discussion is really important, because it is your chance to make your study stand out and as I to make your study shine. So to really demonstrate the relevance and the importance of your work, and to show how it advances the field. So I always like to think of the discussion as my playground. It's my chance to make the most of my results and to put my findings in context. And a good discussion needs to synthesize the results of your study with published evidence, and it needs to use that solid evidence to interpret findings. And a really good discussion links the results to topics presented in the introduction. So again, we have context, the knowledge gaps, the research aims, and the hypotheses. We need to be able, the reader needs to see how the discussion links back to these topics in the introduction. And again, it comes back to this idea that our research aims and hypotheses are the absolute essential thing to think about to structure the rest of the paper. When I get, as an editor, when I get reviewer comments back or comments from a handling editor saying this discussion is unstructured, or it's very hard to follow the discussion, or it's usually because the discussion does not refer back to those initial aims and hypotheses, does not refer back to that broad context that was provided in the introduction. And I always recommend to authors that they frame the discussion around their hypotheses and their questions. And this is a really good way of making sure that the reader can see those links and can understand how your study or how your results have addressed your study aims. So the discussion really is the essence of the paper. When I read papers, I tend to skip the methods and the results, and I will go straight to the discussion, because the discussion often tells me what I need to know about the study. So we need to think carefully about what the purpose of the discussion is. What are we giving the reader? Well, it's first of all, interpreting your findings in a wider context. So placing your findings in against the background of what we already know and understand about the topic. So other published results, for example. Then, very important, demonstrating how your results address your aims and hypotheses. Showing how your findings add to existing knowledge. So how do they go beyond previous work? And of course, you may want to highlight knowledge gaps, directions for future work, and obviously demonstrate how your study advances research in this field. So we can think about a series of questions we can ask ourselves before we start writing the discussion. The first is, have you met your research aims? And if not, why not? Then, have you filled any of the knowledge gaps that you highlighted in the introduction? This is really important. Obviously, how do your findings fit into previous research, but how do they go beyond it and add to our understanding? Are there any remaining questions? And have new questions emerged from your work? So have your results shown a new knowledge gap or raised a new question? And then, of course, what is the wider relevance of your results? What are the implications of the study? So these are really, really key questions that you can ask as you're preparing or drafting your discussion section. If you can show all of these things, you're doing a pretty good job. What I should also mention, and this is really important, you do not have to explain everything. And this is something I often find, especially in early career researchers, this idea that we need to be able to explain every single result, even if it was unexpected. And nobody expects you to have the answers to everything. So one way of thinking about this is actually, if I can't explain a result, is this a new question that needs to be answered with a follow-up study? So I'll come back to that in a moment. What I'd like to do first is show you signs of a weak discussion. So these are largely based on my experience as a reviewer and editor, where I've sort of said, you know, nice study, but it is really let down by the discussion, or the discussion does not highlight the merits of the study. And there are four main types of discussion. The first type is what I call the saga, and this is where results are just repeated and then discussed sequentially without putting them in context. And this is actually how I was taught to write a discussion. But the big problem here is that the reader can't see how the results answer the research questions, and they can't see how the work addresses the field. So if I just go through each result in turn and say, this is what we found, this is how I interpret it, we're not synthesizing. We're not pulling our results together to provide a picture and show how we've addressed our aims. So if we think about framing the discussion around our hypotheses and around our research aims and interpreting them and explaining them by reference to previous work in that context, then we already have a much stronger discussion. I'll also, in the second session, I'll talk a little bit about how we might be able to do this, how we might be able to address this issue and actually synthesize results with the interpretation from the literature. Then the second type of weak discussion is what I think of as the detective story. And this is where I don't have an overview as a reader, but I'm given lots and lots of pieces of evidence. And then at the end, the authors tell me what it means. And this is very confusing. If you don't have the required prior knowledge of the study and the results, the reader will get confused. You will lose them. Because a lot of detail that we're giving and the interpretation we might be giving requires context. Otherwise, we can't understand it. And often, if you don't give the key messages up front, then the links between the evidence and the relevance of that evidence is unclear. So in this case, we can use this funneling technique in the discussion to very good effect, starting big, finishing small. So we start each paragraph with a very clear take-home message or a clear indication what this paragraph is going to be about, and then we build in more detail. The third sign of a weak discussion is, and this is another one I see especially in early career researchers, is that the results are only discussed by direct comparison to other published work. And of course, it's important that we acknowledge previous work, and we must acknowledge previous work. We must also say clearly if other people have found similar results, for example, in a different ecosystem. This is very important to interpret our own work. But the big problem is if my discussion only says, oh yes, we found this, which is in agreement with this other study. We found X that is in contrast to the results of another study. Then the reader is directed towards all of these other studies instead of focusing on the relevance of your work. You're highlighting other people's work instead of showing what you've achieved, and the advance to the field is not clear. So it makes the study sound confirmatory. So instead of making these direct comparisons, again, this technique of synthesizing your results with published findings is really important to show how you've advanced the field by filling remaining knowledge gaps and how your results go above and beyond other people's work. So a very good example of this that I see very frequently is where authors will make a direct comparison to another study and say, well, we found something different. So these people did the same study in a different ecosystem, but our findings show that our system is different. And that's it. There it ends. It's just this comparison. And for me as a reader, this is very unsatisfactory because I want to know why there is this difference. And actually being able to show that there is a difference between ecosystems that we need to consider, for example, is already filling a knowledge gap. So these kind of things are important when we're writing the discussion, this idea of relevance and advance. Then the final sign of a weak discussion is obviously where we over-interpret our results or we start getting lost in speculation. And this is what I call the fairy tale because it's unrealistic. And the big issue here is that the reader loses confidence in your work. Often, So over-interpretation is interpretation that relies on variables that weren't measured. So we're starting to speculate, oh, well, this could be because of, I don't know, changes in microbial communities, and I haven't measured microbial communities. So unless I have very strong evidence that, for example, this phenomenon was explained through changes in microbial communities, and that's been shown in a lot of other work, then we're starting to rely too heavily on variables that were not measured in the present study. We can do it to a certain extent, but when most of our interpretation is based on other people's work rather than our own data, then it starts to become a bit of an issue. Mostly because we're not providing sufficient evidence to support conclusions, because ecosystems differ, taxa differ in their responses to treatments or variables, then we might not have very strong evidence. But as I said before, you don't need to explain everything. And if you need to speculate because you don't have the evidence, then you can also actually present that as a new hypothesis or a new direction for future research. So a knowledge gap that needs to be filled by further study. And then you've also demonstrated how your results are advancing the field. So when we think about the structure of the discussion, it's nice to start with a short paragraph that just summarizes the key findings of the study, puts them in context, a statement of whether or not you've addressed or met your study aims, whether you've rejected any of your hypotheses. This can just be a few sentences that provides a bit of a backdrop for the reader to understand what you're going to discuss and why. Then start each paragraph of the discussion with a clear statement of the key findings that you will discuss, ideally referring back to your study aims and hypotheses. And then within the paragraph, present your lines of evidence for the wider relevance, interpret your findings by synthesizing your results with other people's research. What is also important is to give caveats or limitations to the study in context. So personally, I dislike having a separate section of limitations. Some people prefer that, but it's important to remember that if you put your the limitations of the study in a single section, you are drawing the reader's focus to the fact that your study has a lot of limitations. Instead, you want to give your limitations in context and then give the reader confidence that despite those limitations, your results are solid, your results are sound. And you can only do that if you put them in context and not in a separate section. And then, of course, you want to finish the discussion with conclusions about the wider relevance of your results. And here you can think about the way you structured your introduction in reverse. So you started with the global context, you gave the scientific context, and then the general issue the study was going to address, ending with the study. And now you're kind of starting with what you found and drawing conclusions about that wider context, showing the relevance of the study in that wider context. And I'll come to that. I'll come back to this idea of broadening out at the end of the discussion in the second session. Now, one last thing that I really want to mention before we go to questions in the break is that the conclusions should conclude things. I often read conclusions that are basically like a second abstract, so they just summarize the study. So when we write the conclusions, we want to think about what should the reader remember from the paper, why should they care, and what should we do next? So the worst thing you want is that the paper the reader ends the paper thinking, well, so what? I've just wasted an hour reading this paper. What does it all mean? So that's the worst way to end a paper. So we draw conclusions, demonstrating the advance, demonstrating the relevance, rather than providing a second abstract at the end of the paper. And of course, you know, everything I'm talking about here today, even good writing, does not compensate for poor science. We really need to show that our science is robust, that it is evidence-based, and that it advances the field. And with that, I think we can go to the Q&A and see if we have any questions. We have a first question. Could you please elaborate on the topic of caveats and limitations in the discussion session, is the question. So yes, happy to do that. It's hard to really do it without examples, but basically what I always try and do is that in the relevant section, so if a limitation to the study might influence the interpretation or the wider applicability of the results, for example, then I will mention that in the relevant section of the discussion. And I'll say, for example, you know, this study only included 3 species. So the, you know, wider applicability needs to be tested on other species. However, and then you give the reasons why this limitation is not an issue. So in this case, I would say, however, we chose widespread species representing different plant functional types, for example, and therefore, and we showed that all respond in the same way. Therefore, we believe that this is more widely applicable. So in each case, we would give the limitation. In this case, only a few species, but then we would immediately give the reader confidence by saying, however, in our case for our study, we don't think this is a problem. We still think our results are robust. We still think they're widely applicable. And, you know, it would be great if other studies tested this with other species. So in this case, I've used the limitation to give the reader extra confidence in my results, to highlight the fact that we've used different plant functional types, and also to demonstrate that, you know, it's a foundation for future studies. We want to motivate other researchers to investigate other species. Hopefully that answered the question. Okay, so
yeah, I think a couple of people that raised their hands, but maybe you guys can put your questions in the questions box. Yeah, that would be really helpful.
Yeah, I'm afraid I can't see raised hands.
Oh yeah, that's fine. Yeah, because you won't like they won't be able to unmute themselves. So yes, please put your questions in the questions box. Thank you. I think there was another one that came in.
Yeah.
Yeah.
So the next question is, I think this is how long it takes me to write a manuscript. That's a very good question. I think the shortest duration ever was probably start to finish in less than 4 weeks. So yeah, I think a month working on a manuscript is reasonable. During my PhD, that was kind of how long I took to write a manuscript, because I was working on it full time. Nowadays, I don't have that luxury. But basically, I realized that if I follow all of my own guidance that I'm showing you today, I basically use my own slides as a checklist. It makes it a lot faster to write a paper. So using the structuring techniques I'll show you in section 2, remembering these principles for each section of a paper, can actually speed up the writing process, because the hardest part really is creating that good structure and that strong narrative. And if you have that, then the rest of the writing is actually a lot easier, and you waste less time. So yeah, but you know, I have a manuscript I started writing 3 years ago, and I haven't finished. And it's partly because yeah, partly because I already always have many other things on the go, and it's not high priority. But also in this case, I'm not entirely sure what is the key message I want to get across. And so that for me is the biggest block to writing a manuscript. It's not being able to say what the key message is. So hopefully that has also answered that question. Okay, the next question is, the discussion is hard. How or whether I could summarize a model from published papers. Yeah, so in the second session, one of the things I'll ask you to do at home is to find some papers that you enjoyed reading. So not because the language is good, but because the science is really clear, the relevance is clear. It was very easy to understand what the researchers had done, why it was important. And then you analyze those papers. So we'll look at some examples, but this is how I learned a lot of my writing. I really looked at how other people have structured papers, and I looked at papers that I found easy to read and understand, and then I also looked at papers that I thought were really poorly written and hard to understand, and then I used that to work out how to get messages across. But like I said, I'm going to talk about structuring the text in the second session to get messages across. So analyzing papers is great, and you can definitely create kind of your own model to write a good discussion. And it's also with experience in writing, you learn more and more how flexible you can be and how creative you can be and how you can then deviate from that model. So yes, I think using published papers as a model is a good idea. Just don't copy the text. Then the next question is about being repetitive when trying to place work in context in the introduction and discussion. That's also, repetition is also something I'll come back to in the second session, but on the whole, I often find that a lot of when I'm writing the introduction, it really is an introduction to my study. And so a lot of detail and sort of tangential information that doesn't belong in the introduction, I often cut that out and I save it so that it can go in the discussion. So the introduction is there to provide the backdrop and the context and the justification for your hypotheses or your questions. And all the other detail you can save until the discussion, and then you're not repeating yourself constantly. I think that's probably the best answer I have in this case. The level of detail and the focus is different between the introduction and discussion. Then we have, how can I better describe the hypotheses in the introduction? I can't really answer that question without a concrete example. The important thing is that the hypotheses should address the aims of the study. So what is the goal of the study, and how am I going to reach that goal with my hypotheses? And then remembering that they need to be specific and testable. And of course, you should have your hypotheses before you start doing the study. So before you start measuring anything, before you start collecting data, you should already have your hypotheses, and it's worth spending time on those on formulating those hypotheses. It's absolutely fine if you reject your hypotheses. So a lot of people think, oh, I need to find evidence to prove my hypotheses. And of course, by definition, you cannot prove a hypothesis. You can only reject it, or you can find evidence to support it. And this is science. This is how science works. So if you have a beautifully formulated hypothesis, and yet your data don't support that hypothesis, then you discuss the reasons for rejecting that hypothesis, and often that might be even more interesting than finding exactly what you expected. Then we have another question. How to find a scientific question worthy of study? Most of the time I realize that nearly all have been studied and it's difficult to find a good question. Well, here again, so a good research paper will highlight knowledge gaps. So find recent research on the topic and see what they're saying. What needs further study? What are the knowledge gaps in your research field? So read the literature. Go back as far as you can with the literature. You can also read, a lot. I think a lot of people tend to focus very much on their particular study system, and often you can learn a lot by looking at related systems, but the similar topics, and looking at where the emerging questions are. So I think that's the best way to find questions to answer. Then, we have a lot of questions. We might need to come back to some of these at the end so that you actually get a break. Then we have, when I write the results, I will inevitably repeat the contents shown in the figure, but these contents have already been shown in the figure. Well, actually, overlap between the content of the figure and the text in the results is not a problem. What is a problem is if your figures and tables show the same thing, or your results and your discussion repeat the same thing. But actually describing the results or the content of the figure is not a problem. In fact, it can be very helpful. Then we have, should the article be written concurrently with the experiment, or is it better to start writing after all the experimental work is completed? Depends on, I'd say that depends on the person. So I actually usually try and keep the results concurrent. So writing the method, sorry. So writing the methods as I'm writing, as I'm doing them. So as soon as I've completed part of a study, I'll write down the method so I have a record. And often I have an idea of, I mean, obviously I have my hypotheses before I start doing my experiment. I also have a justification before I start doing my experiment. So I might even have an outline of the introduction before I start. Everything else, I would say it's probably better to wait until the experiment has finished. But again, it depends. Some people like to keep written notes throughout that they can use for writing, and that for writing the paper, and that's obviously very good if you have written notes all the way through your experiment. So not leaving all the writing till the end, but the actual paper I would probably leave until the end, because only then do you know, once you have all of your results, only then do you know what your story is. Then a question about acknowledging previous research. So details about acknowledging previous research. So one thing that's important is, you obviously can't acknowledge all papers on the topic. So choose the most relevant ones. Choose the ones that is it's primary research providing evidence, often for the first time, and to use a range. There is a tendency now in science that we only cite papers from the last 5 to 10 years, because that's what's easy to find on Google Scholar, for example, or other search engines. But it's very important to show, to provide reference to the paper that first demonstrated that, that phenomenon. So wide range, the most relevant, and always focus on primary research. So not not repeating, a review paper, for example, to discuss a phenomenon that has been demonstrated through experimental work, for example. In the first session, we've looked at the purpose of the sections of a paper and the paper as a whole, and hopefully you now have a good idea of how to think differently about writing your paper and drafting your paper. So in this second part, I really want to focus on this idea of telling a story, of creating a strong narrative across the paper as a whole, but also in the individual sections, and how to do that. So this second session will focus very much on these tips and tricks and techniques that you can use to create a strong narrative. As a reminder, the two most important things out of the first session that we really need to remember to create our narrative and structure the text. The first is that the research aims guide the writing process. So every section of the paper needs to, or as you're writing every section of the paper, you need to think about, what were my research aims? What are my questions and hypotheses? And how do I demonstrate how my study addresses those research aims as I'm writing? The second most important thing is this technique of funneling, of starting big, finishing small. So going from broad to narrow, from wide context to specifics and details, and going from simplicity to complexity. This technique is essential for good writing. And again, as I said in the first session, it's essential because it gives the reader the context they need at each level to understand the next level of detail or complexity. So we're guiding the reader through the text. And this is very important for creating a strong narrative. So you will hear it again a lot during the second session. And finally, I mentioned this at the beginning, we always need to think about who is going to read our paper. And my PhD supervisor always said, a study has not been done until it's published. And I would expand that and say, a study isn't relevant until it's been read and cited. And ultimately, this is what we're aiming for. We don't just want to publish. We want our papers to be read and we want them to be cited. And writing a paper that is likely to be read and cited by others, that is what journals are also looking for. So if you want to get your paper in a good journal, you need to make sure that it is attractive to other researchers and that other researchers are likely to cite it. So who is going to read our paper? In the very first instance, it's an editor and reviewers. So we need to think about what the editor is looking for and what the reviewers are looking for. Then once it's published, who do you think should read it? Why might they read it? And what are they going to get out of it? So this is all about writing with the reader in mind. The editor will be looking for a good paper that fits to the journal and that is likely to be read and cited by other researchers. The reviewers are already like a test readership. They represent researchers who might be interested in your paper, who work in that research field, and they will be looking for scientific rigor and relevance in most cases. So they want the reviewers will specifically be looking at how your study addresses your research aims. So we're writing with the reader in mind, and this idea of a benefit to the scientific community is quite important because, again, as I said at the beginning, the advance, this is what is going to get the paper read and cited. We cite papers that give us ideas for new research, that answer a question that we needed to answer to create our own studies or to design a new experiment. And so thinking about the benefits we get from reading other people's papers, this is what we need to give our readers. And the best way to do that is by creating a strong narrative, telling a story about our research. And often finding the story is the first problem, because as scientists, we're very focused on generating and collecting data. So it's important to remember that the ultimate goal of a paper is not to present data, unless you're writing a data paper. But most research papers, what we want to do is show the understanding that we've derived from those data. So we collect data, we start processing the data to get information. From that information, we derive knowledge, and then we interpret that knowledge to create new understanding. And this is sort of the scientific process we need to think about, data collection in the field or from the experiment, but actually turning that data or that knowledge into understanding. That's the writing process. So we need to think about writing a story. And of course, I don't mean fiction, but actually just creating a very clear narrative to demonstrate clearly how the study addresses the aims and why it's important. So we're creating our story using evidence to support the study's objectives, to support our approach and our methods, and to support our interpretation. This is very important, that science must be evidence-based, but we still want to try and bring in a little bit of excitement or curiosity for the subject, because this helps other people relate to what we're doing. So why is my study important? Why is it relevant? Why should people read my paper? And this is what we want to try and recreate with our narrative, with our research story. And one of the things that we don't really do in research is think about storytelling. And most stories follow very, very clear structures. So telling stories is the way human beings have passed on information and knowledge and understanding over millennia. So there are stories that have been around for millennia and that we still tell each other. It is an exceptionally good way to communicate because a good story is memorable. And that is exactly what you want. If you think about other researchers reading maybe 20 papers, or if you're writing a grant proposal as well, the reviewers might have to read 10 different proposals. You want your story to stand out, and you want your paper, your proposal to stand out and to be memorable. You want the people to remember your paper for all the right reasons, and you can do that by telling a story. Now, this story structure, OCAR, this is opening, challenge, action, resolution. This is the most common story structure. If, like me, you like action movies, you can pick any kind of action movie that you can think of. It will probably follow this structure. So there will be an opening, what the story is about, and providing the context, what the reader needs to know to understand the story. So in a movie, this might be sort of the background, sort of the opening 5 or 10 minutes, and then comes a challenge. So what do the characters need to accomplish? What does the hero of the movie, what problems or issues does the hero need to overcome to get that happy end? And then there's a whole load of action. So this is what happens to address that challenge. So the hero fighting the bad guys, for example, and then at the end we have our happy end. That's the resolution, and there we're showing what has changed as a result of all of this action. And we can apply this this structure to scientific papers. So if we think about how we want to structure our paper sort of in more broadly, then the opening is the larger issue we're addressing. So the background to that issue, the wider context. The challenge is then those specific knowledge gaps or specific problems that you're going to address and the questions that you're going to answer. The action is how you go about answering your questions, and the resolution is what we've learned from the work, so the advance. And this structure is actually what we use for specialist journals. So things, all of the BES journals, a good paper in the BES journals will use this structure. Journals like Nature and Science use a very similar structure, but it's much more condensed. So this is very well suited to the classic research paper, and I can show you how we can actually map this OCAR story structure onto the sections of a paper. So the opening is our introduction, that's clear, right? We're introducing our the wider issue, we're giving the background, and we're introducing the study system or the taxa. We're then narrowing down to those specific questions. So we're presenting the scientific issue, the research aims, and the hypotheses. That is our challenge. And then there's a whole load of action. So methods, results, and discussion is mostly action, what we did to address that challenge, what we found, and what it means. And the resolution is then largely part of the discussion, but specifically the conclusions. So the key take-home message, what have we learned? What do we get out of this study? Now, what's really important here is that in research, we focus on that middle part, right? The action. We have spent maybe 3, 5, even 10 years, perhaps, carrying out a study. We're very focused on the methods. We're very focused on the results. And of course, we want to show what it all means. But actually, for the reader reading a paper and for the story, the most important parts are not the action. The most important parts for a strong narrative are the opening, the challenge, and the resolution. So what's really important when you're writing is that readers need to be able to understand the paper, even if they only read those 3 parts. And if your story structure is poor or weak, and the information is not in the right place, the reader will misunderstand your story. So the opening, challenge, and resolution are power positions in the paper. Basically, this means the broader relevance of the study. So what is that wide relevance? What issue is the study going to address? And basically, has it addressed it? And what are the next steps? Those 3 things are really, really important. And of course, if you think back to the structure of the abstract, all of those 3 things need to be in the abstract. So that opening, challenge, and resolution, and your action is shrunk down to a single short paragraph in the abstract, maybe just 2 sentences even. So most of the abstract actually focuses on opening, challenge, and resolution. So these are the important parts of the story that will attract readers to your paper. And we need to make sure that they're in the right place. So if we think back to our classic 4-paragraph introduction again, we have why the topic is important in the broader sense. So that's basically our opening. Then we have the narrower scientific context. So the opening is narrowing down towards the challenge. This is all opening. The key knowledge gaps that we're going to address, that's our challenge. And of course, the project's novelty, how it addresses the knowledge gaps, how it's going to advance research. There we're already promising our resolution. So you can see that in both the abstract and the introduction, we have this OCR. So these 3 really important parts of the story. And we need to give these in the right order. So if we put our specific challenge right at the beginning of the introduction, the reader will not understand that it's the challenge. The reader will read it as an opening. These things are maybe a little bit abstract, but I'm going to show you in more detail how we can achieve this structure: opening, challenge, resolution. And the most important thing to remember at this point is that of course the action is important, and of course we need to describe it, and we need to do so very clearly. But when we're thinking of creating a narrative, it's this opening, challenge, resolution that is essential, that we need to get that right as well. Then I talked in the first session a little bit about this widening out again in the conclusion section of the paper, that we need to link back to the issues we've shown in the introduction. And this is basically creating a balance between opening, challenge, and resolution, and it creates what we call an hourglass structure in the paper. So the opening of the paper, the introduction, gives us the wider context, narrows down towards the challenge, and then the discussions and conclusions show how the challenge has been addressed and widen back out again, because the resolution links our conclusions to the wider context of the paper. And this is quite important, because if your opening is too narrow, people will misunderstand how relevant your study is. And if your opening is too broad, when people get to the resolution and realize that you've overpromised in a way, you've promised too much and not delivered, they will feel disappointed. So getting this balance between opening and resolution right is quite important. Then how do we actually get from opening to challenge to resolution? Well, there are these things called story arcs. And again, it's a very abstract concept, but essentially story arcs are bridges. And they get the reader from the opening through the action towards the resolution. And again, here, the opening, the resolution, or the beginning and the end of an arc are the power positions. So if you think about a bridge, how important it is that the bridge has very solid foundations on either end, story arcs are the same. So story arcs create a clear flow of information and logic throughout the paper. Now, this is quite an abstract technique, so I'd actually like to show you a few examples of how to create story arcs across the paper, but also in individual sections. Because essentially, your paper is one big story arc, but you can actually create story arcs within each section, within each paragraph, and even to some extent in single sentences. Now, to create story arcs, there are a few, I guess, general principles or rules. So the first thing is that each unit should make a single clear point. So for your paper, you might have one big aim, one clear take-home message. That can be broken down into 2 or 3 key results, 2 or 3 hypotheses, but usually your paper should make a single clear point. And in the rare instances where as an editor, I've asked authors to split a paper in 2, and that does happen, it's because the paper does not make a single clear point. It's because there are 2 stories that have been mixed up, and it's very, very confusing, and it's very hard to create this structure. Then, of course, each section has a single clear point there defined. So obviously introduction, methods, results and discussion, but also each paragraph is a unit, a unit in itself. So each paragraph each subsection, each paragraph needs to make a single clear point. And of course, sentences are a unit of thought. Then we want to link the sentences within a paragraph, and we want to link the paragraphs within a section. And if we can do that, we know we have a nice red line going through the manuscript. We have a strong narrative that is easy to follow. And I'll show you how to do that in a moment. Having these story arcs and thinking about these links between sentences within paragraphs and paragraphs within a section helps us identify extra material that breaks the arcs. And this is particularly important for those of you who asked about creating a narrative and how to structure the discussion and reduce repetition. You can use these story arcs to identify when you have something that doesn't fit, because you won't be able to link the sentences within the paragraph, and you won't be able to link the paragraphs within a section if you have something in the wrong place. Then, because each of these units should make a single clear point, you want to resolve old topics before you introduce a new one. This is particularly important in paragraphs. There is an increasing tendency to write little mini paragraphs. So people see a big block of text and find it scary and think it's going to be hard to follow a big block of text. And actually, when you're writing in English, a paragraph is on a single topic, and you shouldn't artificially split it just because it's too long. So old topics in paragraphs, for example, need to be resolved before you introduce the next one. And you can think of this like the sections of a paper. You write the methods in the method section, and when the method section is finished and you go on to the results, you don't start describing new methods. It's that simple. It's the same with individual paragraphs. But you can link paragraphs to create a nice arc. So, simple tricks for creating arcs. Start each paragraph with a topical sentence. So a sentence that gives sort of the topic of the paragraph up front in the first sentence. Then you successively build in detail and complexity, so you use this concept of funneling to support that topical sentence. You build up complexity and detail, and then you end the paragraph either with a statement that clearly resolves the topic. So, this topic is resolved, I'm going to move on to the next. Or you can end the paragraph with a statement that links to the topical sentence of the next paragraph to create a lovely arc throughout the section of the paper. And if you're very good, you can do both. So I'm just going to show you an example. Again, I realize that the text is very small here. So all I've done is color each individual part of the paragraph, and you can see a description on the left-hand side. We have in this paragraph a topical sentence. The topical sentence is, in the Arctic tundra of Alaska, plant growth is limited by nitrogen availability, especially in the tussock community. So this single sentence tells us that we're going to be talking about limitation of plant growth by nitrogen availability. And we also know what our system is. It's the Arctic tundra of Alaska, and we're probably going to be studying the tussock community. Then the next sentence gives general information to back up that statement. So here it says, even where soil nitrogen content is relatively high, nitrogen availability is low because cold temperatures and waterlogged conditions slow the decomposition of organic matter, and so on. So here we've basically backed up our topical sentence. We've provided a little bit of extra detail to explain why plant growth is primarily limited by nitrogen availability. Then the authors have used funneling to build in more detail about the specific processes. And then they've even used funneling to build in complexity. So we've gone now from plant growth in general, nitrogen availability, we've explained why, we've given the processes that are involved in decomposition and the accumulation of organic matter. And then we've brought in the complexities caused by climate change, that warming can increase soil organic matter decomposition in this case, and therefore release nitrogen and enhance plant growth. The implications of these impacts are then given, and then at the very end of the paragraph, we have this resolution. And the resolution is, as a result, nitrogen supply to plants is one of the primary factors regulating the carbon balance of the Arctic tundra. So essentially, this resolution actually reinforces the topical sentence. So explains why this topic of nitrogen supply to plants is so important. It's important for the carbon balance. We're linking back to the Arctic tundra. And then the authors have done something lovely. The topical sentence of the next paragraph is then about nitrogen mineralization. So they say net nitrogen mineralization has been considered to be the principal control on plant nitrogen availability. So what they've done here is that they've started a new topic, nitrogen mineralization, but they've linked it to this concept of the first paragraph, which is plant nitrogen availability. So this is a really lovely, very complicated text, actually, or a lot of different concepts are introduced within one paragraph, but they're very clearly linked. And the whole paragraph is funneled nicely, it's resolved, and then it links to the next paragraph, in this case, of the introduction. So this is a nice example of really strong arcs and how to create these really strong arcs within a paragraph using this funneling technique. Okay. So one way of doing this is actually to use a sort of a condensed version of this OCAR story structure. This is the LD or LDR story structure, where we can use it to create arcs on a small scale. So this is basically just lead, development, possibly resolution. And in this case, we have a very brief opening, and the challenge comes fairly quickly. And then we give lots of additional information, funnel that information, building in details and complexity. And often this development can actually be merged with the resolution. So we don't have to have a separate resolution. And this structure is much faster paced than the OCAR structure. It tends to be what is used in papers like Nature and Science, but it's also really suitable for individual sections, for paragraphs, and for sentences. So what we saw in the previous example, so in this example, there is no action here. There is just this lead development, and the challenge is already sort of implied in the text without, the resolution is, the challenge is there in the text, it's implied in the text, and the resolution is more resolving the topic and finishing the paragraph rather than resolving the challenge, if you like. So here we have this lead development structure in the paragraph. So we can use this structure and the funneling technique to create these arcs within paragraphs. The first sentence, this is the lead. So our topical sentence tells us what the paragraph is about. And then the body of the paragraph provides additional information that's necessary to understand why this topic is relevant in the context of our study. And that's exactly what we've just seen in this example. Then, of course, you can use that final sentence of the paragraph to provide a sort of a conclusion to the paragraph or link to the next paragraph. So this is a really good way of using a story structure or thinking about how you want to structure your paragraph and use that funneling technique to create a really good, strong narrative. That's all there is to it. I'd like to show you an example of what happens when it doesn't work. So this is a real example. I've anonymized it and I've taken out citations, but this is a real example. This example is by native English speakers who normally write very well, and I think, in fact, I know that this paper was written in a hurry. And here there is a big problem with this paragraph because it's full of broken arcs. And I'll show you why. If I color the text, basically I've just colored the text by subject or by topic. So my paragraph starts, light is considered the most limiting resource in the understory of tropical forest. So this is my topical sentence. It indicates that the paragraph is about light. But then the text continues, one model of nutrient limitation. So suddenly it's not about light anymore. So we've started with light and then we've got nutrient limitation. Then the next sentence starts talking about nutrient addition, which is actually another topic. It hasn't been linked properly to the nutrient limitation idea. And then we go back to nutrient limitation. So back to the blue. Then we go back to light. And then at the end, we go back to nutrient limitation. So you can see we're jumping around among topics. There is no narrative. There is no clear thread throughout this paragraph. And I can show you this in more detail if I break down these individual statements. They really are individual statements jumping between topics. And you can see the topical sentence indicates that the paragraph is about light. But then the resolution at the end of the paragraph, we have this sentence, we investigated nutrient limitation of seedling growth. So the study is actually about nutrient limitation. So we can actually restructure, we can take this information and we can restructure the paragraph to make it make more sense by linking these different colors together, putting the different colors together in the paragraph. But actually, in this case, we probably want to create a second paragraph that is about light and not about nutrients. So this paragraph deals with several different topics and jumps around from one topic to the next. And that is a broken arc. This is what I'm talking about when I talk about broken arcs. And it is very, very confusing to the reader if you have a topical sentence about light and then most of the rest of the paragraph is about nutrients. And it's also very confusing if you keep jumping between topics. So you have one sentence on one topic, the next sentence on another, the next sentence on another. So again, each unit should make a single clear point, and we want to link each unit in a way that creates a nice strong narrative. So I've shown you how to identify an arc or broken arcs. And what I'd like you to do, so I mentioned in the Q&A session that it's really useful to identify, to analyze papers, to understand these techniques and see how people apply them. So I'd like you to choose 2 papers. Ideally, you know, when you finish the session or tomorrow, when you're back at work, choose 2 papers, one that you really like, one that is easy to understand, easy to follow, and then one that isn't, one that is really hard to read. And then have a look at the introduction of your papers and see if you can find the arcs or the broken arcs. So finding the arcs, does each paragraph make a single clear point? Have the authors linked the paragraphs within a section? And if so, how have they done it? Are there some of these broken arcs? Is there information there that breaks that flow of logic where you're not sure why they've introduced this topic, or why, you know, what does this have to do with the rest of the paragraph? Anything that puzzles you or makes you wonder why information is there. And then also have a look at how the authors have actually resolved each topic before they introduce the next one. Have they linked paragraphs? Have they provided a more definitive resolution? These techniques are really important for creating a narrative. And I can almost guarantee you that the paper that is easy to understand has clear story arcs. So has units that make a single clear point, units that are linked well, and topics that are resolved before the next one is introduced, or very clearly linked to the next topic. The paper that's harder to read and harder to understand will be more like this example, where the authors jump around between topics. The topical sentence doesn't actually tell us what the paragraph is about, and there isn't that clear flow of logic throughout the text. So analysing papers in this way is really important, and do try this with 2 papers. Try it with more, but definitely really have a look at some papers that are easy to read and some that aren't. You'll find it isn't necessarily a language issue. It is a structural issue of the text. And this is really important because it means that if you can get the text structure right, if you can create a strong narrative and a well-structured text, then language issues don't matter as much. And there were a couple of questions in the Q&A about using AI for scientific papers. And, you know, at the moment, this is a very hot topic, but this is one way you can use AI to write your papers, in that you create your structure, you use your own words, and then you just use AI to correct the language. And in fact, we even offer that through the BS journals. We offer language editing service through Writeful, where you can upload your entire manuscript and get language editing online. So this is one really good way of using AI ethically to help you write your paper. So you do the writing yourself, you create that story and that structure, and then you use AI just to correct the language. So I've talked a lot about paragraphs and sections, but actually sentences also have a structure that we can use to best effect. So we can use even sentence structure to help create a strong narrative and a strong paper. So again here, the start of the sentence provides the topic. Then we put any additional information in the middle of the sentence, and then the important information is emphasized at the end of a sentence. And this is called the stress position. This is really, really important. It's a very important technique in writing English because of 2 things. First, if there is anything you want to highlight or emphasize, you know it has to go at the end of the sentence, not at the beginning, and definitely not in the middle. And if there's anything you don't want the reader to focus on, you put it in the middle. Okay, so this is called the 3-1-2 sentence structure. So by order of reading emphasis, the reader puts most of the emphasis on the last part of the sentence, the second most emphasis on the first part of the sentence, and least emphasis on the information in the middle. And you can use this to your advantage. For example, when I was learning to write, my supervisor said, "Oh, you know, put the main result, like the big significant result, at the start of the sentence, and this non-significant result at the end." And it's the wrong way around. If you start a sentence saying, you know, we found that, you know, I don't know, our treatments had significantly increased soil carbon storage, but this had no effect, whatever it is, had no effect. The reader is going to focus on, oh, this had no effect. If, however, you say, although this treatment had no effect, our other treatment significantly increased soil carbon content, you have that non-significant result to provide the topic, but the emphasis is on that significant result at the end of the sentence. So this is a really powerful technique, something to remember. Then we can use the sentence structure to create arcs. So if we think about what we've been looking at so far, a good story has a beginning, a middle, and an end. And then, of course, we have our lead development resolution. We've got our opening and then challenge action resolution. And we've also got topic and stress. So in each case, we're thinking about three parts, so beginning, middle, and end. And we can use those to really good effect. For example, coming back to limitations and caveats of the scientific study, you know, if you're going to give limitations, put them in the middle of the paragraph, not at the beginning and not at the end, because that way you're being upfront and you're stating the limitations, you're being completely honest about the limitations, but you're putting them somewhere where the reader will not highlight them or emphasize them. Okay. The same goes with things like so-called negative results or non-significant results, and that was also a question in the Q&A. How do we, you know, should we put negative results in a paper? And for me, the answer is always yes. So there will often be many things in a study that don't work, and unless it's because of a flaw in the study, I do think they should be reported alongside the significant results. It's much harder, obviously, to publish a paper that is only negative results, where nothing really worked, unless you can really demonstrate that the study was very robust and rigorous. But actually reporting negative or non-significant results alongside the significant ones is really important for interpretation, and it's a service to the scientific community, because if you don't write about it, nobody knows you've done it, nobody knows it didn't work or it wasn't significant, and then they may waste time and effort designing a study to test something that has already been tested and failed. So hopefully that also answers that question. But these non-significant results can go sort of in the middle or at the end of in the middle of a section, in the middle of a sentence, or you might want to give them at the start to provide the topic, and then finish with the important, relevant information, the significant results. So I've got two sentences here. We've only got this one poll in this session. But I'd like to know from you, if you read, I'll read these two sentences out. I'd like to know which sentence works better for you. So which one do you think has a stronger arc? So sentence one is, by increasing physiological stress, drought reduces soil microbial activity and builds up and causes a buildup of biodegradable carbon that is rapidly respired upon rewetting. Sentence two Drought reduces soil microbial activity by reducing diffusion and increasing physiological stress, causing a buildup of biodegradable carbon that is rapidly respired upon rewetting. So please vote. Which sentence do you think is the stronger arc? Which one follows the principles I've just been talking about? 1 or 2? Let's have a look. So 69% for number 2, 31 for sentence 1. That's interesting because it's actually if you're not sure what you're looking at or looking for, it's actually quite difficult to differentiate between these two sentences. But I'm going to show you why sentence 2 has the stronger arc. So here we go. So sentence 2, the topic is really clear. I'm talking about drought. The first sentence starts by increasing physiological stress, and drought comes much further on in the sentence. So it still has that topic, but the topic is quite weak. What is clear in both sentences is that the important information is this concept of carbon being rapidly respired when soils are re-wet. So that's common to both sentences. But the second sentence gives a very clear topic. In the middle of the sentence, we have the additional information, again, to support that topical part of the sentence. So drought reduces soil microbial activity. That's our topical statement. And then we have that additional information that we don't really want the reader to focus on. It's not our emphasis, but we are explaining that initial statement. So drought increases physiological stress and causes this buildup of biodegradable carbon. So in this case, we have our classic 3-1-2 sentence structure, most important information at the end, the topic at the beginning, and everything else in the middle. Now, why is this important? I can show you this if I expand and add another sentence. So now we have that first sentence. Drought reduces soil microbial activity by increasing physiological stress, causing a buildup of biodegradable carbon that is rapidly respired upon rewetting. The next sentence reads, the sharp increase in respired carbon immediately after rewetting is referred to as the Birch effect, and the strength of the Birch effect is often related to the frequency or severity of drought. Now, I've coloured topic and stress here so you can see what I've done. I've basically linked the topic of my new sentence to the stress of the previous one. And this is an absolute certain way of creating strong arcs throughout a paragraph and making sure you don't have tangential information that breaks your story arc. So you don't have to do this for every single sentence, but you should be able to do it for every sentence in the paragraph. If you can link topic and stress all the way through the paragraph, you know you have a strong narrative and a really clear story arc within your paragraph. And it's the same then with the paragraphs within a section. If you can link the topic of one paragraph to the stress of the resolution of the previous one, so the topical sentence of a paragraph links to the resolution, the last sentence of the previous paragraph, then you know that you've got a strong story arc at that level, so across the section or even across the paper. Okay, so this is a really, really powerful technique, linking topic and stress, linking lead or opening and resolution. So now I can add another sentence to introduce a new topic. In this case, now I'm talking about frequency and severity. So stronger and more frequent droughts are predicted under numerous climate change scenarios. So now I'm actually introducing something new. I'm linking this frequency and severity of drought to climate change scenarios. And you can imagine that my next statement is going to be something about climate change. So you can see how this builds up a really nice narrative and a really nice flow in the text. So it's a very, very powerful technique. So what I'd like you to do at home with your two papers is to also analyze them to see whether they've done this. Have they used topical sentences in their paragraphs? Where have they placed the most important information within the sentences and paragraphs? And are the sections, paragraphs within, or the paragraphs within the introduction discussion, are they linked? And what about the sentences within paragraphs? How much have the authors linked these sentences? And I'm sure everyone will be aware, if you've read any scientific paper, you will see words like however, nonetheless, nevertheless, those kind of things. These are alternative ways of linking sentences within a paragraph. They do the same job as linking topic and stress, and also create a strong narrative. And they're the ones that are easy to misuse. So you can use, often I'll see something where however or not, is used, for example, to introduce an entirely new topic that doesn't link to the previous one. And that's a mistake. You really need to use these link words to the best effect to create that strong structure and strong narrative. Okay. So I want to move on to this idea of the resolution. There we go. Sorry, there's some information missing there. So I want to move on to this idea of the resolution because it's actually often misunderstood and quite important, especially when you're hammering home those key points of your paper. So first of all, I want to show you how not to conclude your paper. So weak resolutions, for example, I see often in the conclusions or the end of the discussion or at the end of the abstract, the authors tell me that this is really important, but they don't tell me why. They don't tell me how it's important. They don't tell me what the advance is or how, what are the consequences? What are the implications? Then another weak resolution is that there are unanswered questions that are just not addressed. I don't know why they're not answered. And sorry, this is the wrong way around. The knowledge that's been gained from the study has not been synthesized into new understanding. So I've been presented with a lot of facts, but my understanding of the topic has not increased. Then there are distractions. So again, often in the conclusions, just generalizations, just existing information or a summary, which distracts me from the importance of the paper, or new information is introduced for the first time. So anything that is in the conclusions must have been discussed. Otherwise, you're introducing new information, and that is confusing to the reader. And then the most common way, or most common mistake I see in the conclusions is that suddenly the limitations of the study are described, especially if they're described for the first time. Sometimes reviewers will require it, but if the reviewers require you to put limitations of the study in the conclusions, always follow with how those limitations show us what to do next. What are the studies we need to achieve? What are the questions we need to answer that would address those limitations? And of course, the most common one is that papers conclude with further research is needed. And essentially, this downgrades your work. It undermines your work because you're telling the reader that you haven't done enough work, that your research is not enough. And that's a really bad way of ending a paper. So instead of that really general broad statement, further research is needed, instead you want a clear statement about how your results tell us what kind of research is needed. So what do we need to do next based on the findings of your study? That's a good way of resolving your paper. So when you're writing the conclusions to your paper, you can write, you want a strong resolution, and you want to achieve this hourglass structure of the paper, balancing your opening and your resolution. And you can do that by working backwards through from your resolution, action, challenge, and opening. So you're actually highlighting the key message of your study first, the resolution, showing how you got there and how it links back to that broad opening, how it links back to that wider issue and contributes to addressing it. Obviously, you want to highlight the achievements of your study and show how it will generate new research. And you want to conclude with emerging scientific questions that arise directly from your findings. So a good resolution should actually sort of complete the circle, linking back to the opening of your paper, and obviously show how the research has advanced the field, how it has contributed to our understanding of the topic. Okay, so that's really all I wanted to say about structuring the paper to create a strong narrative. One way of finding this resolution and making sure that it links back to the opening is quite simply to write your brief summary of the key results and then synthesize them. So pull them together to show how they answer your question, and use that to then demonstrate to the reader how your study has met its aims and contributed to that wider issue. I do want to talk briefly about this idea of synthesizing, and I spent a lot of time thinking about this because it's very hard to demonstrate how to synthesize information, but it's a very important writing skill. And synthesis is quite simply combining different aspects of your ideas in your research with the ideas of others, so previously published work, to produce something new. So in this, I've got this example here. We're taking lots and lots of different ideas, our ingredients, and we're turning those separate ideas into a nice tasty cake. So we're combining one thing with another to form a new whole. We're integrating information. And this is a really important writing skill. You need to use it in a literature review, because you need to demonstrate in a literature review common patterns and emerging ideas from lots and lots of individual studies. In the text of a primary research paper, which is what we're talking about today, you can synthesize the results of previous work to provide context in the introduction, synthesize the results of different measurements or experiments to show how the study has answered research questions. And of course, then in the discussion, you're integrating your results or synthesizing your results with other people's published findings to provide context and interpretation without distracting from your work, from your study. So synthesis is quite an important skill, and it's very hard to teach and quite hard to understand. I'm going to skip over this. This is a way to get started with synthesizing. So thinking about sources, organizing notes, starting to connect ideas, and then thinking back about how it will support your case in the most brief description possible. But have a look at this. What I would like to do is just very briefly show you some issues with synthesizing results and how to address those issues. So these are common problems that I see, for example, in a discussion in this case. And I talked about this earlier, that we often get, we'll often just repeat results and then give an interpretation. So, and I'm also guilty of this. I always reread my discussion and edit it afterwards because I realize I do this constantly. Here's an example. So basically the issue is that in the discussion, we repeat a result and then we give an interpretation from the literature. So we found that literature from pioneer tree species decomposed faster than litter from pioneer tree species decomposed faster than litter from old growth forest species. That's our sentence, our result. Then previous research by these authors has shown that pioneers have leaves with high nitrogen content, which represent a valuable resource to decomposers. High nitrogen content could therefore explain our result. So, as a starting point, this is fine, but this is not synthesized text. And essentially, what we can do is break this text into bullet points. We have 3 completely separate statements. So if you see this issue with your writing, then you know you have not synthesized the information properly. And there are 2 solutions to this. The first one is to rephrase the explanation and put it first. So now my text is, the high nitrogen content of pioneer tree species represents a valuable resource to decomposers, which explains why litter from pioneers decomposed faster than litter from old growth forest species in our study. So suddenly I've taken those 3 statements, I've synthesized them into a single sentence, which explains our result. And also you can see by the structure here, I started with the topic, high nitrogen content of pioneer tree species, and I've ended that sentence with our results. So I've used this sentence structure. Topic is previous research, and the stress is our study. The second solution is to rephrase the result and just combine the sentences. So here I've started, it is likely that litter from pioneer tree species decomposed faster than litter from old growth forest species because pioneers have leaves with high nitrogen content, which represent a valuable resource to decomposers. So here there's it's a slightly different way of rephrasing the results. Both are perfectly valid, and which one you choose to do obviously depends on what the next topic is going to be. So how are you going to continue this discussion? If I'm going to the first solution basically resolves that topic, or it could also continue with the discussion about old growth forest species and how they decomposed. In the second solution, I'm probably going to talk about decomposers next. So you can see how I can synthesize results simply by rephrasing them, and then how I do it depends a little bit on my narrative and flow. What you also see here is that the synthesized versions are much more concise. So 1 sentence in each case, basically, instead of, was it 4 previously? And the second is that in natural sciences, we always give citations in parentheses unless we actually want to emphasize a particular study or particular authors. So normally we would give the citations in parentheses. A second brief example is also very common. An example from a literature review we have here. These authors showed that because of high temperature variability in temperate forests, soil temperature plays a significant role in controlling greenhouse gas fluxes. So there we have our main statement. But in a Danish spruce forest, soil CO2 efflux was strongly correlated with increasing soil temperatures. In addition, changes in soil temperature caused most of the temporal variation in CO2 fluxes in Austrian beech forests. This is very common, and the issue here, and the reason we know that this is not synthesized, is that this is like a list of individual studies. So if the text reads like a list of specific examples, then you haven't synthesized the information well. And the solution to this is obviously pulling those 3 studies together to show the common pattern. So now we have soil temperature plays a significant role in controlling greenhouse gas fluxes in temperate forests. That's our topical sentence. And of course, using funneling rules, we now provide extra detail to back that sentence up. Due to high temporal variability, soil CO2 efflux increases with rising soil temperatures, and changes in soil temperature cause most of the temporal variation in CO2 fluxes. So here I've cut out all of that detail. We don't know where these studies took place. We do know they're all in temperate forest. And again, the citations at the end, because I'm not drawing attention to single studies, I'm demonstrating a big idea or a common pattern. So synthesizing information often requires cutting out a lot of detail. What is important here is that I can use specific examples. For example, if I really want to emphasize an important point, or I want to support the statements with a specific example. So synthesizing information doesn't mean I can't give examples, but it means I will use them to best effect to emphasize a point rather than listing lots and lots of examples. So that's really it. That's all I wanted to say about synthesizing information. Like I said, again, you can look at your papers and see how other people have done it. How have they linked previous research to their own results? Has it been done well, or can you separate statements into like a list and see how well it reads and how compelling the narrative is? So just to summarize, we can think about synthesizing information about integrating different ideas and different results to create something new. And in each case, when we're writing, we are working towards addressing clear aims and clear questions. So thinking about the purpose of the sections of the paper, how everything links back to our study aims and our hypotheses can help with synthesis as well as narrative and structure. Then when we're synthesizing, we're using connections, relationships, or patterns rather than individual examples, but we use case studies or individual examples to demonstrate or clarify complex concepts or provide additional support or evidence. So overall, to summarize sort of the very key points, or the crucial points of the second section, is thinking about how a good paper is a story. It follows a clear story arc. It has a beginning and a middle and an end. We keep it simple, so single clear point that we're trying to make. Obviously, it needs to be concrete and it needs to be credible. And we do that by providing evidence from our own data and other people's work. A good paper synthesizes the knowledge we've gained from the study into new understanding based on that evidence, and it concludes with strong take-home messages that demonstrate an advance to the field. And with that, hopefully, we can spend a little bit more time on questions. Hopefully, I've already talked a little bit about or answered the questions about the use of AI in science papers. What I would like to add more generally is that you need to be quite careful using AI because of plagiarism. We're seeing more and more papers where there's a lot of overlap with previous publications, and basically AI will just search previous texts online and recombine them into whatever you tell it to do. So first of all, AI is only as good as the instructions you give it, but also there is always going to be a danger that what you're given is actually copying text from somewhere else. And that gets flagged up when you submit your manuscript. So my advice for using AI is to always make sure you're doing your own work, doing your own writing, putting everything in your own words, and if you need to, use AI to correct your own work, but not to actually use it for writing. That would be my advice at the moment. And of course, you should be upfront about the use of AI as a tool. I think our general policy at the moment is that it should be that should be flagged up in the cover letter or in additional information. Is that correct, Rowena?
So yes, if you use AI as part of your methods in any way, then that should be in the methods section as Emma covered. But if you use it in any way to edit the text, then you should put that in your acknowledgements section, just to acknowledge that you've used, for example, Writeful to edit the language and improve it. And you're not going to be penalized at all for using programs like Writeful to edit your language, but it's just good to have that honest and upfront.
Thank you. Great. So then hopefully I've already addressed the question about how to better organize the discussion into a story and a narrative. So again, framing around the study aims and the hypotheses is always a good thing to do. Linking back to those hypotheses, linking back to the initial questions, and then using these techniques, the funneling and story arcs to make sure that there is a really nice, strong narrative through the discussion. So hopefully that is answered. Then somebody has asked, I always modify the finished draft paper. I always think it should be written better. And that's slow progress. How should I solve this? If you can write a perfect draft paper in one go, you are an exception. I know very, very few people who go through less than, say, 5 drafts of a paper, even before they're ready to send it to co-authors. So papers, what you see, the finished, polished product, has been through several drafts, co-author comments, reviewer comments, editorial comments. The vast majority of us cannot write perfectly the first time. What you can do is create an outline to make sure you have your narrative, and you can use the techniques that I've shown you to shorten that time, so to make the writing a bit more efficient, because you will always have much more work if you don't have a strong structure and a strong narrative. If you have a strong structure and a strong narrative using these techniques, then the additional comments will be a lot easier, or the additional revisions or edits will be smaller and a lot faster. And again, when we conduct research, we may design many factors to test a hypothesis, but in most cases, most of the factors are invalid. If you mean invalid, then if it's completely invalid, then it's not worth discussing. If they're just non-significant, so you have clear expectations, clear biological reasons or ecological reasons to test these factors, but they turn out to be non-significant, then you should discuss them, but you won't go into depth. So you can present them, and you might want to discuss why you didn't find a significant result, but what you really want to discuss in depth is anything that is an important advance or something that is really relevant. It can be a negative result, but usually it's more about what you did find. Then there's a question about writing a little bit every day. Yes. So I think this is great. It's really good to get into the writing habit. It doesn't have to be working on a paper. Most of us are asked to write literature reviews, for example, while we're working on experimental work. We might want to write a proposal for a grant, you know, even if it's like a travel grant or some research support funding. And actually, even if you don't have any of that and you don't really have writing tasks, reading a paper, reading a few papers every day, and making notes of anything that might be relevant to your study, that can help you massively with writing the paper later. So it helps you get into that flow of writing, gives you practice, but it also gives you a really useful collection of notes when you do start writing the paper. Writing chunks of materials and methods, absolutely. I always encourage my students to do that. Simply because you need to have a record of what you've done. You may as well write it as you would write the methods to a paper. And obviously collecting information that you can use for the introduction, discussion, and interpretation while you're conducting your study can actually help you think about the patterns you're seeing in your data and the results you might get at the end. Yes, there's a question about papers like the Nature stable of journals require the results and the discussion to be written together. And then there are some journals allow either separate sections or together, and how to determine which format is suitable. I find writing them together harder. And it's the hardest thing is actually writing them separate and then trying to integrate them. I've actually just been working on a paper where the postdoc did exactly that. So wrote for one journal and then decided with the classic structure, separate results and discussion, and then decided to actually submit to another journal where they're written as a single section, and it was very, very messy. So it's really hard to do after the fact. In general, I find writing results and discussion separately easier personally. And when you've got a choice, and obviously when it's where you submit is a completely different question, I guess. So if you're submitting, you know, how to choose a journal requires a lot longer than we've got today. I think the most important point is you really need to choose the journal based on its aims and scope. So whether your paper really addresses what that journal is looking for and what they want to publish. I think that's the most important part. And again, I think it's personal preference whether you find it easier to write results and discussion together or separately. It'll depend a little bit on how comfortable you are and what type of study it was. Oh, and then we have the last question, AI and data analysis. I'm not really, this goes beyond writing the paper, but so yeah, a lot of people are using AI for coding, for example. So asking AI what statistical tools, how to formulate a model, for example, how to write R code to test a specific topic. And again, that I think that is absolutely fine as long as you do the groundwork, so you know what question you need to ask. You know the suitability of your data set, because AI can't tell you that, I mean, unless it sees your data. But asking, you know, I have these data sets, this is what I need to test, what I want to use this model. So knowing which statistics you want to use, and then asking AI to help you with writing the script, that's, I think, perfectly acceptable. And I know a lot of people who do it. But again, the ideas, how you do it, understanding the suitability of your data and the suitability of the statistics, that is absolutely essential for the correct use of AI to then help you with the actual coding. So it's very similar to the writing. You need those original ideas. You need to understand what you're doing. You need to know which questions to ask. And that is the most important element that needs to come from a human being. Thank you, everybody, for attending. I really hope that was useful.
Thank you, Emma. Thank you so much for doing this, and thank you everyone for joining.
Great. Thanks, everyone. Bye.